Bismillahirrahmanirrahim. So today we are going to study about the lab diagnosis of Escherichia coli. In the previous lectures, we have already discussed about the infections caused by the Escherichia coli, like the watery diarrhea, the bloody diarrhea, the urinogenital tract infections like the cystitis, pyelonephritis, and the systemic infections like the sepsis and the neonatal meningitis and their pathogenesis. We have already also discussed the epidemiology and introduction of the Escherichia coli. In this lecture, we will study about the lab diagnosis of Escherichia coli. So kindly watch the other lectures of Escherichia coli as well. So the first thing, in order to diagnose Escherichia coli, what we do gram staining. So on gram staining, what we will find straight gram positive rods. Straight gram positive rods. So on gram staining, we will find straight gram positive rods. The second things which will help in the diagnosis of Escherichia coli is the culturing. And the most common culture, if we suspect Escherichia coli, is the use of McConkey agars. And what does this McConkey agars do? It differentiates the non-lactose fermenter from the non-lactose fermenters. So if it is lactose fermenters, we will find pink smooth colonies on the agar. But along with Escherichia coli, there are also many bacteria which are also lactase fermenter, and they also give this pink smooth colony. So in order to differentiate this Escherichia coli from the other bacteria, we will do some other tests. And the first one is the first differentiating test is the eosine methylene blue agar test. We will use the eosine methylene blue agar, and in this we will find small plate colonies having green metallic sheen, and this small plate colonies having green metallic sheen is characteristics of the Escherichia coli and the eosine methylene blue agar. I will try to show some images to you. So now here you can see the McConkey agars. This is the pink smooth colonies formed by the lactose fermenters, and these are the non-lactose fermenter colonies having yellow color. And this is the eosine methylene blue agar, in which you can see plate colonies having a green metallic sheen. This is the characteristics of Escherichia coli. The other test with the help of which we can differentiate the Escherichia coli from other lactose fermenters is the indole test. And indole test is positive for the Escherichia coli because it can convert tryptophan into the indole. The second one is the use of acetate as its carbon source. So Escherichia coli is the only lactose fermenter which can use acetate and not citrate as a source of carbon for its energy production. The other lactose fermenters use citrate as a source of carbon for the energy production. The third one is the lysine decarboxylation. This Escherichia coli can decarboxylate lysine, while other lactose fermenter cannot. And the fourth one is the motility, that this Escherichia coli are usually motile and the other lactose fermenters are non-motile. So the first test is the first differentiating test is the usine methylene blue agar test. The second one is the indole. The third one is the use acetate and not citrate. The third one is the lysine decarboxylation, and the fourth one is the motility test. We can also do some other tests like the TCI test, triple sugar iron test. And triple sugar iron test, we can find that there is that the Escherichia coli produce acid on slant and butt, and this is mainly due to lactose fermentation. There is acid both on slant and butt, and it is mainly due to the lactose fermentation, because this lactose fermentation will produce acid, and there will be acidic slant and butt, i.e. there will be the change in color of the phenol red into yellow at both slant and red. I will try to show you the picture. The fourth one, the D1, it is the acid at both slant and butt. So such kind of test tube will you see of the Escherichia coli and the TSI test. And we can also do the methyl lead test, in which there will be acid production due to the glucose fermentation. As we have already discussed that Escherichia coli is an enterobacteriaceae, and all the enterobacteriaceae ferment glucose for its energy production. So this was the some points regarding the lab diagnosis of the Escherichia coli. Also we can do some serological tests and other DNA based or RNA based tests like the PCR, etc. But in our book, in the Levinson microbiology, these were the some tests, or these were the some cultures in microscopy which we were discussed. So I have only focused on that, not on the clinical lab diagnosis. Now coming towards the treatment, that how do we treat and what are the drug of choice for the disease caused by the by the Escherichia coli. So the first one is the diarrhea. So we do not use any kind of antibiotic usually, because they are mostly, the watery diarrhea is mostly self-limited. So we mostly concentrate on the rehydration. So they are usually self-limited, so you may mostly concentrate at what? On the rehydration. The second one is the hemorrhagic aerobic syndrome, and in this also we do not use the antibiotics, because the antibiotics are contraindicated in the hemorrhagic uremic syndrome, as we have already discussed, contraindicated. Keep in mind this point, because this antibiotic will try to kill the or break down the bacteria, and there will be much more increase in the concentration of sugar toxin, and it will complicate the scenario. The third one is the neonatal meningitis. So in order to treat neonatal meningitis, we use the combination of ampicillin plus cefotaxime. Ampicillin, this is the ampicillin, and this is the cephalosporin. Ampicillin plus cefotaxime uses the combination of these drugs for the treatment of neonatal meningitis. The fourth one is the cystitis. So for the treatment of cystitis, we mostly use the oral trimethoprim, sulfamethoxazole, or the nitrofurantoin. These are the two drugs of choice for the cystitis. And for the pyelonephritis, we mostly use ciprofloxacin or ceftriaxone. For the pyelonephritis caused by the Escherichia coli, what we use the ciprofloxacin or the ceftriaxone, third generation cephalosporin. And for the treatment of sepsis sepsis, we gives the antibiotic like the third generation cephalosporin or the aminoglycoside. Third generation cephalosporin like the ceftriaxone or the aminoglycosides. So these were some points regarding the treatment of diseases caused by the Escherichia coli and the lab diagnosis of the Escherichia coli. So that's all for today. Thank you.